BEST is a suite of motif-finding programs that include 4 motif-finding programs: Align ACE, Roth et al., 1998, Bioprospector Liu et al., 2001, Consensus Hertz and Stormo, 1999, MEME, Bailey et al., 2006, and the optimization program BioOptimizer, Jensen and Liu, 2004. BEST was compiled on Linux, and thus it can only be run on Linux machines, Che et al., 2005. SeqMotifs is a suite of web-based programs to find regulatory motifs in co-regulated genes of both prokaryotes and eukaryotes. In this suite, Bioprospector, Liu et al., 2001, is used for finding regulatory motifs in prokaryote or lower eukaryote sequences, while CompareProspector Liu et al., 2002, is used for higher eukaryotes. Another program in Scan, Liu et al., 2002, is used for finding protein DNA interaction sites from ChIP on chip targets. These programs analyze a group of sequences of co-regulated genes so they may share common regulatory motifs and output a list of putative motifs as position-specific probability matrices, the individual sites used to construct the motifs, and the location of each site on the input sequences. CompareProspector has been used for identification of transcription factors MEF2, MI, SRF, and SB1 motifs from a human muscle-specific co-regulated genes. Additionally, in a C. elegans C. Brixit comparison, CompareProspector found the PHA4 motif and the UNC86 motif. Liu et al., 2004, another C. elegans CompareProspector analysis showed that intestine genes have GATA transcription factor binding motif that was later experimentally validated, Pally et al., 2006. The Regulatory Sequence Analysis Tools, RSAT, is an integrated online tool to analyze regulatory sequences in co-regulated genes, Thomas Challier et al., 2008. The only input required is a list of genes of interest, subsequently upstream sequences of desired distance can be retrieved and appended to the list. Further tasks of putative regulatory signals detection, matching positions for the detected signals in the original dataset can then be performed. The suite includes programs for sequence retrieval, pattern discovery, phylogenetic footprint detection, pattern matching, genome scanning and feature map drawing. Random controls can be performed with random gene selections or by generating random sequences according to a variety of background models, Bernoulli, Markov. The results can be displayed graphically highlighting the desired features. As RSAT Web Services is implemented using SOAP and WSDL, so either Perl, Python or JavaScript can be used for developing custom workflows by combining different tools. TFM, Transcription Factor Matrices, is a software suite for identifying and analyzing transcription factor binding sites in DNA sequences. It consists of TFM-Explorer, TFM-Scan, TFM-Value and TFM-Cluster. TFM-Explorer, Transcription Factor Matrix Explorer, proceeds in 2 steps. Scan sequences for detecting all potential TF binding sites, using JASPAR or TRANSFAC, and extract significant transcription factor. TFM-Scan is a program dedicated to the location of large sets of putative transcription factor binding sites on a DNA sequence. It uses position weight matrices such as those available in the TRANSFAC or JASPAR databases. The program takes as input a set of matrices and a DNA sequence. It computes all occurrences of matrices on the sequence for a given p-value threshold. The algorithm is very fast and allows for large-scale analysis. TFM-Scan is also able to cluster similar matrices and similar occurrences. TFM-Value is a software suite providing tools for computing the score threshold associated to a given p-value and the p-value associated to a given score threshold. It uses position weight matrices such as those available in the TRANSFAC or JASPAR databases. The program takes as input a matrix, the background probabilities for the letters of the DNA alphabet and a score or a p-value. RVista, Regulatory Vista, combines searching the major transcription binding site database TRANSFAC Professional from Biobase with a comparative sequence analysis, and this procedure reduced the number of predicted transcription factor binding sites by several orders of magnitude. Lutz and Ovcharenko, 2004. It can be used directly or through links in Vista, Genome Vista, or Vista Browser. Human and mouse sequences are aligned using the Global Alignment Program AVID, Bray et al., 2003. Mulan brings together several novel algorithms, the TBA multi-aligner program for rapid identification of local sequence conservation, and the multi-TF program for detecting evolutionarily conserved transcription factor binding sites in multiple alignments. In addition, Mulan supports two-way communication with the GALA database. Alignments of multiple species dynamically generated in GALA can be viewed in Mulan, and conserved transcription factor binding sites identified with Mulan/multi-TF can be integrated and overlaid with extensive genome annotation data using GALA. Local multiple alignments computed by Mulan ensure reliable representation of short and large-scale genomic rearrangements in distant organisms. Mulan allows for interactive modification of critical conservation parameters to differentially predict conserved regions in comparisons of both closely and distantly related species. The uses and applications of the Mulan tool through multispecies comparisons of the GATAsgene locus and the identification of elements that are conserved in a different way in avians than in other genomes, allowing speculation on the evolution of birds. Motif voter is a variance-based ensemble method for discovery of binding sites. It uses 10 most commonly used individual basic motif finders as its component, Wijaya et al., 2008. Align ACE, Hughes et al., 2000, Meme, Bailey and Elkin, 1994, Bailey et al., 2006, ANSPEC, Mitra, Bioprospector, Motif Sampler, Improbazor, Space, InDScan and Weeder. All programs can be selected individually or collectively. Though the existing ensemble methods overall perform better than standalone motif finders, the improvement gained is not substantial. These methods do not fully exploit the information obtained from the results of individual finders, resulting in minor improvement in sensitivity and poor precision. ConSite is a web-based tool for finding cis-regulatory elements in genomic sequences, Sandelin et al., 2004. Two genomic sequences submitted for analysis are aligned by ORCA method. Alternatively, pre-aligned sequences can be submitted in Clustal, MSF, GCG, FASTA or pairwise BLAST format. For analysis, transcription factors can be selected on the basis of species, name, domain or user-defined matrix, raw counts matrix or position weight matrix. Predictions are based on the integration of binding site prediction generated with high-quality transcription factor models and cross-species comparison filtering, phylogenetic footprinting. ConSite, Sandelin et al., 2004, is based on the JASPAR database, Portales Casamar et al., 2009. By incorporating evolutionary constraints, selectivity is increased by an order of magnitude as compared to single-sequence analysis. ConSite offers several unique features, including an interactive expert system for retrieving orthologous regulatory sequences. Opossum identifies statistically overrepresented, conserved TFBSs in the promoters of co-expressed genes, Hosuai et al., 2005. Opossum integrates a precomputed database of predicted, conserved TFBSs, derived from phylogenetic footprinting and TFBS detection algorithms, with statistical methods for calculating overrepresentation. The background data set was compiled by identifying all strict 1-to-1 human/mouse orthologs from the Ensemble database. These orthologs were then aligned using ORCA, a pairwise DNA alignment program. The conserved non-coding regions were identified. The conserved regions which fell within 5,000 nucleotides upstream and downstream of the transcription start site, TSS, were then scanned for TF sites using the position weight matrices, PWMs, from the JASPAR database, Portales Casamar et al., 2009. These TF sites were stored in the Opossum database and comprise the background set. Toucan2 is an operating system independent, open source, Java-based workbench for regulatory sequence analysis, Aerts et al., 2004. It can be used for detection of significant transcription factor binding sites from comparative genomics data or for detection of combinations of binding sites in sets of co-expressed/co-regulated genes. It tightly integrates with Ensemble and EMBL for retrieval of sequences data. Toucan provides options to align sequences with different specialized algorithms. Visavid, Bray et al., 2003, Lagan, Brudno et al., 2003, or BLAST. Motif scanner algorithm is used to search occurrence of sites of transcription factors by using libraries of position weight matrices from TRANSFAC 6, Mattis et al., 2003, JASPAR, PlantCare, Lescott et al., 2002, SCPD and others. Motif Sampler can be used for detection of overrepresented motifs. More significantly, Toucan provides an option to select cis regulatory modules using the module search. In essence, Toucan 2 provides one of the best integration of different algorithms for identification of cis regulatory elements. Web Motifs Web Server combines TOMO and theme tools for identification of conserved motifs in co-regulated genes, Romer et al., 2007.